Imagine stepping into a long-sealed tomb, a frozen glacier, or an arid desert where ancient bodies lay perfectly preserved for millennia. Here, the evidence isn't days old, but thousands of years. How do scientists recover whispers of DNA from these incredible human mummies? From the 5,300-year-old Ötzi the Iceman to the regal remains of Egyptian pharaohs like Tutankhamun, without destroying the very history they seek to uncover. It all hinges on meticulous collection and extreme care. Welcome back, future molecular time travelers. In this episode, we're diving into archaeological trenches and donning biohazard suits, exploring the extraordinary lengths paleogeneticists go to collect DNA evidence from the ancient past. Our focus today is on how we retrieve this precious genetic information, especially from remarkably preserved remains. Ancient DNA isn't found just anywhere. Its survival depends on specific environmental conditions like cold, dry, or low oxygen environments, which slow DNA degradation and allow for natural mummification. Paleogenetics studies DNA from an incredible range of ancient life forms. We collect from human remains, like dense bones or valuable mummified tissues, natural or embalmed, which gave us the DNA of Ötzi the Iceman from his icy tomb and the Egyptian pharaohs from their arid burial sites. But our scope extends further. We find DNA in animal remains from permafrost, like intact woolly mammoths, and in ancient plant remains from sediment cores. We even extract microbial DNA and environmental DNA, eDNA, which holds genetic traces of entire past communities without needing physical remains. These diverse finds truly show what ancient DNA can reveal about the deep past. Handling ancient samples, especially fragile mummified tissue or bone, presents a critical challenge. Modern human or environmental DNA can easily contaminate and overwhelm the tiny ancient signal. Preventing this is the paramount goal in paleogenetics. This means adhering to clean room principles from the moment of discovery. Paleogeneticists wear extensive personal protective equipment, PPE, like sterilized suits and multiple layers of gloves. All tools are new, sterile, or meticulously decontaminated. They establish sterile work zones even at excavation sites. Sample collection protocols are incredibly strict. Minimally destructive sampling, often removing outer surfaces of bones to eliminate contaminants, and prioritizing material from undisturbed contexts. Meticulous documentation and collecting reference samples from all researchers are vital safeguards, ensuring the integrity of these ancient whispers. Once collected, these precious ancient samples, whether delicate mummified remains or fragments of ancient plants, embark on a carefully controlled journey from the field to a specialized, often purpose-built, ancient DNA laboratory, a true fortress against contamination. This involves secure, sealed, and often frozen transport to maintain sample integrity and prevent any further degradation or contamination during transit. Upon arrival, these labs themselves are engineering marvels designed for sterility. They are often in separate facilities or entirely isolated sections of buildings, far away from modern DNA work. They utilize positive air pressure, meaning air flows out of the lab, preventing outside contaminants from entering. UV light sterilization is regularly used on surfaces and in the air. A strict unidirectional workflow is enforced. Samples only move from dirty pre-PCR areas to clean post-PCR areas, never backward. Even reagents, plastics, and equipment are dedicated solely to ancient DNA work. The initial handling in the lab involves careful surface cleaning to remove any residual dirt and environmental contaminants before the actual DNA extraction process begins. Every step is a calculated move to protect the integrity of the ancient genetic samples. From the frozen tundra to the desert tomb, from the bones of early humans to perfectly preserved ancient plants and animals, collecting ancient DNA is a testament to scientific rigor and patience. Every recovered fragment is a victory against time, a potential key to understanding the deep past. But simply collecting the DNA isn't enough. Next time, in Part 3, we enter the shared molecular battlefield. exploring the universal lab techniques from specialized PCR power-ups to next-generation sequencing that both forensic scientists and paleogeneticists use to read those tiny, degraded whispers of DNA. You'll see how the same molecular tools unlock secrets from both crime scenes and ancient history. Make sure to subscribe to The Geneticist, like this video, and ring that bell to catch all our deep dives into genetics. See you soon.